Hello viewers, welcome to RJ's Botany. I'm back with the new video on topic DNA as genetic material. Michel discovered DNA in 1869 and it was established that chromosome chemically consisted of proteins and DNA, and it became clear that protein consisted of 20 amino acids and DNA is made from 4 nucleotides. By scientific experiment, it was clear that protein having a more varied and complex structure and could be possible a genetic material. So the British microbiologist Frederick Griffith provided the first evidence for DNA as genetic material. First of all, we have to know what are the characteristics of genetic material. Genetic material means it was considered as DNA as genetic material. The characters are it should inherit from parents to offspring. It ମାନେ ଯାହା କ୍ୟାରେକ୍ଟର୍ସ ଥିବ ପ୍ୟାରେଣ୍ଟସ୍ଙ୍କ ପାଖରେ ସେଇଟା ଛୁଆମାନଙ୍କ ପାଖକୁ ଟ୍ରାନ୍ସଫର୍ ହେଉଥିବ, ଇନହେରିଟ୍ ହେଉଥିବ. Second point is it should have the ability to control or change one particular trait of the individual. And as DNA contains gene, so gene has the ability to control a particular character of an individual. So DNA was verified as genetic material through two experiments. One is bacterial transformation experiment, which is a transforming principle, and other one is the bacteriophage infection experiment. Here we are going to discuss about transforming principle or bacterial transformation experiment. This experiment was performed by Frederick Griffith. By experimenting with a pathogenic means a disease causing bacterium Diplococcus pneumoniae, that is known as pneumococcus. This bacterium causes pneumonia in man and most mammals and has two phenotypes. And those two phenotypes are, one is virulent means pathogenic form and other is avirulent or non-pathogenic form. The first one, that is S strain. S means smooth, or it is a virulent or a pathogenic bacteria and form and which possess a polysaccharide coat that protect the bacterium from phagocytic attack of the host. That means the bacteria has capsule on the shell wall, and because of that capsule coat, the virulent bacteria form smooth edged colonies in culture. So these virulent strain or S strain causes pneumonia. And the other one is virulent or R strain means a rough, non-virulent or avirulent, non-pathogenic strain. And it lacks coat means the bacteria lack capsule and it is harmless. They form rough edged colonies in cultures. The virulent forms are therefore called smooth type or S type, and the avirulent forms are rough type or R type. So R strain do not cause pneumonia. And there are several types of strains of S strain and R strain like S1, S2, S3 and R1, R2 and R3 etc. But Griffith selected mouse as the host and S3 and R2 bacteria for his experiment. So Griffith used mouse as experimental animal. Coming to the topic here, Griffith experiment. Griffith injected A strain means smooth strain bacteria, which is a virulent bacteria, to a mouse, and mouse died of pneumonia. Means Griffith कौन कले ना इंजेक्ट कले A strain जोटा virulent bacteria virulent strain सेटा को कौन कले इंजेक्ट कले माउस को, तापर माउस देखिले कौन रिजल्ट ना माउस डाइड ऑफ pneumonia. Then he injected R strain bacteria to another mouse and found that mouse survived. ଯେତେବେଳେ H strain bacteria ଦେଲେ ମାଉସକୁ ସେତେବେଳେ କ'ଣ ହେଲା, ମାଉସଟା ମରିଗଲା, କିନ୍ତୁ ଯେତେବେଳେ R strain ଦେଲେ ସେତେବେଳେ ମାଉସଟା ବଞ୍ଚିଲା, survived. In the next step, he injected some heat killed H strain bacteria, that is S3, and he found that mouse survive तामाने जेतेबेले से heat kill estrone bacteria दउचंदी सेतेबेले mouse भी survive कोचे and in the next step he mixed R strain with heat kill estrone से कौन कले R strain ना heat kill estrone को मिसेले and inject to a separate mouse आउटे mouse को देले तो he found that the mouse died from pneumonia तो एठी conclusion से कौन पाइले ग्रिफिथ जेतले A strain देले सेतेबेले pneumonia होची mouse मरुची R strain जेतले जोचंदी mouse बंचुची जेतले heat kill estrone दउचंदी सेतेबेले mouse टा बंचुची but R strain और heat kill estrone जेतले दउचंदी सेतेबेले ताको pneumonia होची and mouse died. So here Griffith thought that something passed from heat kill estrone bacteria into estrone bacteria through a liquid medium. So from heat kill cells of ACE3, something would have escaped and transformed the R2 to ACE3. So this was referred as transforming principle. But Griffith was unaware of the nature of transforming principle. He could not find any causative agent for this transformation and called it as transforming principle. So this is all about transforming principle or bacterial transformation experiment or Griffith experiment. Hope you like this video. Thank you.